All right, moving stuff across the membrane. We've got facilitated active and bulk transport. Facilitated passive diffusion. Facilitated passive diffusion. So this is a protein-assisted diffusion where you've used these membrane proteins to actually allow for movement of stuff across the membrane of the cell. Very classic examples are amino acids and glucose. Glucose, why is it so important? Because the cell primarily is going to live on glucose. That's an energy source for the cell. So, another clinical example, your diabetic patient. Okay? More and more and more of you are going to be treating diabetic, unmanaged patients because it's on the rise. That's fact. In all the medical publications and scientific publications, we are not seeing a decrease in the incidence of diabetes. We're seeing an increase in the incidence of diabetes in every population. Okay? Every socioeconomic group, every ethnic group, it's happening across the globe. So you have a meal. You have an In-N-Out burger, right? That's coming. We talked about Chick-fil-A. We're going to have, equal marketing representation in this class. So In-N-Out's coming. You have a meal. You have your fries, right? They're animal style, whatever you like. Okay, you've got your chocolate shake. Maybe you're a strawberry shake person. After that meal, you have a tremendous amount of blood sugar floating through your body. If you can't figure a way to get that sugar into the cell, your cell cannot use it for metabolism to make ATP. And that is functionally the problem with diabetic patients. There's plenty of sugar in the blood. It just needs to be inside the cell. You've got to move it inside the cell through a carrier molecule. Insulin is the primary mechanism in order to do that. But there are other ways to do it, like with GLUT4. Okay? In fact, GLUT4 is a very potent sugar transporter. And exercise upregulates the insertion of GLUT4 in the membrane of muscle cells. And that's one of the reasons we tell our diabetic patients, you should exercise a little bit more. Because guess what? It's a non-insulin mechanism to get sugar into the cell and out of the bloodstream. Right? Helps your heart, does a lot of great things. But a little bit of exercise in our unmanaged diabetic patients, that's why we say those things. Okay, so let's look at these. These channels, otherwise known as pores, you can kind of see them here. This is the extracellular fluid, the ECF. This is the intracellular fluid, the ICF. And we've got molecules that want to move from outside to in. The solute can bind into the binding site. A conformational change will spit it out on the inside process, right? It's very, very simple. It's kind of like a gate. And nerve cells operate this way. In fact, this mechanism, you can put a star next to this. This is actually how we're going to look at action potentials later in the semester. We're just going to move sodium and potassium through channels like this and allow for an electrical signal to be sent. So with active transport, we've got the movement of solute against the concentration gradient. It requires energy because energy input is needed in order to move the opposite direction of the concentration gradient. It's like rolling a ball uphill. Roll a ball downhill, you just drop it, let it go. No energy is required. To push it uphill takes energy. So these proteins are sometimes called pumps because they take energy, like a pool pump or a water pump. And these pumps, the primary transport is what we call a sodium-potassium pump. And it pumps how many sodiums out of the cell? 3. Very good. How many potassiums into the cell? 2. Okay, you remember this. This is such an important pump, the sodium-potassium ATPase pump. We're going to continue to use it, and hopefully you're not so sick of it. You'll see it not just on a couple of more slides here, but you'll see it throughout the semester. But if we look at this slide, active transport, we can put a sodium-potassium pump here on the basal side of this cell. This is the apical side. Basal side down here, apical side towards the top. Focus on the bottom first. Sodium-potassium pump. Seeing ATP, you cleave adenosine triphosphate. How many phosphate groups? 3. Every time you cleave a phosphate group, you liberate energy. So we go ATP to ADP, that's adenosine diphosphate, that's only 2, and you have a free phosphate hanging out here. You've just cleaved the phosphate, releasing energy. You do this to move a high concentration of sodium outside the cell. Now look at the top. You have high sodium outside the cell. You can bind it to a glucose. Sodium's going to come in down its concentration gradient and carry glucose with it. Does that make sense? That is secondary active transport. Secondary active transport, the glucose hitches a ride down the sodium concentration gradient, and you burned your energy down here on the basal surface with the sodium-potassium pump. Kind of cool. So the sodium-potassium pump maintains a resting potential where you've got higher potassium inside the cell. So higher potassium inside the cell, higher K plus inside the cell, back to our periodic table. Higher sodium outside the cell, Na plus. Both of them are a single positively charged ion. So in cold weather, like here in Flagstaff in the wintertime, thyroid hormone stimulates cells to produce more sodium-potassium pumps. And as these pumps run, how many of you ever put your hand on a pump that's running? Is it what, cold or warm? It's warm. This actually helps to establish our basal metabolic rate and generates heat energy during the wintertime for us. And your thyroid gland actually upregulates your sodium-potassium pump insertions in your cells during cold weather. 30 to 50% of all ATP in the body runs these pumps day and night. Pretty important. All right. Last slide of content, bulk transport. Sometimes macromolecules are too large to fit down some of these channels or these transporters. So we'll use macromolecular transport through what we call exocytosis and endocytosis. Exo, like EX, like exiting. Exocytosis is spitting it out of the cell. Endocytosis is actually bringing it into the cell. So you can see in a picture here, this is an example of how cholesterol is brought in to a cell if it's in the bloodstream. So you've got, on the far left, you've got the receptors that bind the molecule. The membrane itself invaginates or forms a pit. It pinches off and internalizes that molecule, known as endocytosis. If they're small molecules, we have a term for it. We call it penocytosis, which actually translates to cell drinking. And phagocytosis, like macrophages that are part of your immune system, they do this when they're gobbling up bacteria from an infection. Phago is the root for eating. So penocytosis is cell drinking, phagocytosis is cell eating. That's how you bring stuff in through macromolecular transport. Questions? Okay, great. Don't forget, I'll send out an announcement. Quiz 2 and 3 are due on Friday by 11:59 p.m. Make sure you're here next week for some participation stuff.